Professor P. Raveendran, Vice Chancellor of Calicut University. Then, along with Professor P. Venugopalan, again a chemistry professor from Punjab University as well as from Calicut University. Then Professor Dr. Rajendran Vasudevan, again he was in the inaugural session, biomedical research, and Dr. Grinson George, Director, ICIR, Central Fisheries Research Institute. I request all the dignitaries to occupy this. So the criticism can be soft.
Dear chairs and students, scientists, passionate scientific ideas pursuers, I am here to present my ideas on murburn concept. This talk is about the broad outlines of claims, terms, context and impact. In fact, you can rephrase the title, how do we live and why do we die? I am a professor at Kasaragode. With my thanks already mentioned in this talk, I will not be mentioning thanks. So let's get on with it. I started this murburn presentations first with the Parent Institute of Science in India at IISc, and that slide and poster was made by Vivian, who is sitting there, challenging Peter Mitchell's theory. And then we conducted 2-day workshop at IIT Mumbai, and then in 2024 we had a 3-day symposium at Amrita. This is the next in the series. It's a 2-day discussion forum. This discussion forum was a brainchild of actually our Murburn sir, sitting in the front there. I believe that science should be discussed and debated, not just one person comes, speaks and just walks away. The format is all of you sitting there. If you ask good questions, you get prizes, and there is also going to be quizzes for the students at the end. Alright, so please pay attention. Before I proceed, our Shivkumar sir has already mentioned some of the ideas that I have already got to say. Now, scientific temperament, all of the professors and teachers and seasoned professionals talked about the critical spirit that science is pursued. Science is just one of the facets of human endeavor. We, as humans, we believe in loyalty, art. There are so many other codes of pursuing science. But when we have science, the basic idea is, if you try to, the purpose of science is to explain reality or natural phenomena. So imagine you have a statement made that an ant goes in a circular path. You take points and you try to plot them, and if it agrees with the circle's equation, you say that it's okay. It obeys by that theory. But then you can have points here, and all that, and yet it might not be close to reality. There could be a few points, and it need not be a continuous phenomenon also, but you can still fit the process to a circle, disregarding some aspects of reality. You have to explain, the theory should explain all aspects. And when you come to the math, if these are the experimental points, you can fit the mathematical fit with different types of equations. They all might give statistical analysis, correct? And what happens is, usually when a theory has a limited scope, let's say it can explain only this volume, what we do is insert a cone into that, and all incompatible observations or findings you try to put in that. So basically, this is got to do with how science is pursued. And when we try to explain this image by Escher, Escher's waterfall is very, very important. Explanation is about, not about math, not about equations. It's how we try to understand something. So, if you see this image, you see from the second floor water falls down, it turns on the turbine, and then it pushes the water all the way. At any particular point, if you see, the artist has made it impossible for us to discern. But from our natural experience, we know that this is highly unlikely. It is not possible. So, we have to understand that when we have to say that something is right, we need infinite points to say something is right. But to say something is wrong, we need only one point. It doesn't abide by the circle. So, in science, validation or ratification is secondary. In science, critically questioning and refutation or falsification is primary. So, the scientific philosopher Karl Popper said, scientific advancement fundamentally rests on the premise practice of empirical falsification. So, falsification, refutation is key. So, in science, claims, evidence, arguments are important. Of course, consensus is also important, but that is a secondary aspect. It can come later. Alright? So, this is the philosophy of science that I shall pursue. And Mr. Shivakumar already said, advances one funeral at a time by Max Planck. And it's very interesting to note that Max Planck opposed the quantum electrodynamics and other quantum physicists that came. These people opposed, even Einstein opposed. But later on, they became accepted. So, here is one thing that I want you to think about, send your key. Dionysians and Apollonians, both are interesting and important. Dionysians, like me, they make radical changes, but then systematic improvement and expansion of known fields, that's also important. So these are the fundamental aspects of science that need to be considered, and the role of critics in the advancement of science. So, like I said, claims, evidence, argument. So first I state my claims. The claims are murburn models establish continuum of physics in biology. Physics is the mother of all sciences. Math is the language of it. And in murburn models, completely revamped the most fundamental reaction on planet Earth, that is photosynthesis, which gives us oxygen that we breathe and the food that we eat. Then each cell goes through respiration. That process also, then how body produces heat, how you see, how you process information, how intelligence, sensing, you name it, murburn is involved. Metabolism, drug metabolism, epigenetics, post-translation modification, pharmacokinetics, pharmacodynamics, ecology, mechanophysiology, how bacteria move, toxicity of cyanide, immunology, origin, sustenance, termination, evolution of life. What Professor Raveendran was talking about, your Miller experiment. This is more aligned with that. So fundamentally, what I claim is the most fundamental aspect of life is impossible to be explained without invoking murburn concept. That's a tall claim to make, I know fully well. But I hope at the end of these 2 days, or at least this talk, you will see some substance in it. So, what I say is, we know that living beings, the reality has got, we try to understand reality by physics, and that has got basic foundations in information, then space-time and quantum theorization, and fundamentally life we try to study here, at this level, the world of objects, biology, chemistry, and physical physics. In this, I shall argue that murburn concept bridges the domain of non-living from physics. Life emerges through murburn. So, murburn concept, central dogma, that is DNA gives RNA, RNA gives proteins, catalyze all that, then cell theory, cells are the fundamental units of life. So, the organization of life, if you need to understand how powering, coherence, homeostasis, electromechanical activity, sensing response, without invoking murburn, you cannot. This is a fundamental claim of my talk. So, before I go into the details. I would like to tell you. Each talk in this meeting will be focusing on first, what are the classical views? What are the things that you learn in textbooks? Then, what is wrong with those ideas? Briefly. And then, what is the murburn model? And then, comparison. That's how we are going to do it. It's up to you to take it out here. Alright? So, the classical is cells are the fundamental units of life. I'll quickly go over this. DNA codes for proteins, and proteins have beautifully shaped active sites which can bind to various molecules, ions, all other kinds of components that are involved in cells. So, the selectivity and specificity of reactions are governed by the genes that code for the proteins which have a particular active site, and that can distinguish. I know Professor Raveendran looks different from Dr. Grinson or Professor Vasudevan because I know their topology has registered the space, I can map it, I differentiate. Similarly, at a molecular level, the catalysts, those agents that transform reactions, they fundamentally identify molecules with this mechanism of topology. So, this lock and key active site or induced fit theory of identifying is a core aspect of control in biological reaction. Professor Vasudevan asked, how do you control in murburn? So, we come to that. So, one other aspect of the classical theory is very highly deterministic membrane pumps and channels. The cell functions through generating electric gradients or chemical gradients, and they are harnessed by some kind of molecular level intelligence, and pan multiphasic orchestration is also solved by localized microorganisms. So this is what it is, whether it is enzyme substrate reaction, electron donor acceptor, or receptor ligand. So serial sequential and multi-phase spatial coordination requires this theory. Okay. So next aspect is powering, the most fundamental aspect. Now we know a phone or this projector, it has some printed electronic circuits, and you apply a potential which is a driving force. Now our body is a soft matter, fluidic. We don't have possibilities for wiring like we have in these systems. So, but the classical system, whether they are mitochondria, the powering units of the cell, they say that, like what Professor Vasudevan already mentioned, NADH, after food is broken down, ultimately goes through glycolysis, Krebs cycle, and that which makes NADH. Then NADH, through the electron transport chain, the protons are pumped out, creating a gradient for this idea, Peter Mitchell got the Nobel, and then the gradient is dissipated through complex 5 or ATP synthase, and this idea got rotary ATP, which goes round and round to churn out ATP, just like our mills we have. So these are all Nobel recognized ideas, and then these are the fundamental ideas for physiology advocated by Cambridge School of Thought, the British ones, and taken over by the rest of the world. That is, a cell's membrane is kind of a sentient sentinel composition after 2 days. If I eat 2 aspirin tablets and it doesn't go out of my body, then I will cease to be worried. I will be worried plus aspirin, right? So that is not acceptable. So all this is classically central dogma. That is, genes give proteins and active sites, they control everything through active site chemistry. That's the classical theory. Now let's come to the limitations of the classical. What do you think are the simplest things that even 10th or 12th class students can understand? Fundamentally, a mitochondrion, which is considered the powerhouse of this cell, because given a Nobel Prize, it is got very low volume, and the express concentration is very low, and if you multiply it with the Avogadro number, you should get the proton number. So practically, mitochondria or cyanobacteria, they are practically aprotic. There is no proton to be pumped. And if you imagine proton can be formed from water, this is a highly endergonic process. So ATP synthesis The ATP bond itself is only 36 kilojoules per mole. Water is even stronger. So, there is no way Peter Mitchell's proton pumping can work because mitochondria is so small that protons cannot be available freely. This is 10th class chemistry. So when I sent this to the Nobel laureate who is still alive in UK, he said, yes, you have done something. I cannot refute it, but let the world decide. So this is first point. Second point is, we know basically that when the membrane is based in a particular way, the potential, positive, negative charge like that, you cannot have ions going crisscross at their own fancy. There should be some rationale. And also in cyanobacteria, you see, it can carry out both photosynthesis and respiration, and one requires water breakdown to form the nicotinamide reduced agent, and in the other, it requires nicotinamide reduced agent to form water, and in both processes, ATP should be formed. And in this electron transport chain, the idea that Sir was talking about, it requires some common elements. How will cytochrome know that I have to do photosynthesis now, I have to do respiration now? It is not possible by this serial kind of idea. And most importantly, how can things avoid this inevitable chemistry where oxygen can easily gain an electron to make superoxide? It's highly exergonic, very facile. So oxygen would shunt every step. It is not possible to get that wiring. So fixed EM polarity, electrons and protons cannot go in an unrealistic deterministic mandate is yet another fundamental argument. Third argument, Stephen Hawking made one very beautiful example in Brief History of Time. If you take a hot cup of tea and you drop it, the tea cup breaks, the heat dissipates, the water spreads. You cannot have the same process that is highly spontaneous. You cannot have it reverse, but you can still make a hot cup of tea. Or else, you take a juicer, you put fruit from the top, you rotate the juicer clockwise, you get fruit and pulp at the bottom. Now, you cannot say, I rotate the juicer in the other way, I invert the, the juicer, and I put fruit, juice and pulp, and I get fruit in another way. That's impossible. That's not possible in the real world that we live. There are reactions that work when the delta T is very small. You by concentration changing, you can, by flux manipulation, you can have. But these kind of ideas of mechanical work, rotary work that spans multiple spaces, all that, this is just anthropomorphic extensions. They have very little foundations in reality. And most importantly, these are things that even a school-going kid can understand. First and foremost, an erythrocyte, it doesn't have nucleus. It doesn't have mitochondria. It sustains for 3 to 4 months happily. How can it? The glycolytic pathways cannot have millimolar levels of ATP. It is impossible. And also, the control of the cell by genetic regulation, upregulation, downregulation, that's not possible because there is no DNA. And cyanobacteria, as I said, is too small. One of the biggest problems is a proton is 0.00002 angstroms. And a hydrogen atom is 2 angstroms. Imagine trying to control, specifically bind 0.00002 angstrom by a 2 angstrom minimally small atom. It's impossible. These ideas have very little foundation in reality. So, aprotic environment, photosynthesis occurring side by side, proton being too small, and most importantly, the classical theory says that 3 sodium is pumped out and 2 potassium is taken in by this Gen schools who got a Nobel for that, sodium potassium pump. But then plants don't have such pumps. It is known. And then plants yet have 10 times more potassium than sodium. But the problem is potassium is a larger ion. Sodium is a smaller ion. So if you have a smaller ion inside, we can accept it through some kind of sealing. But we have the larger ion of potassium, greater ion. So the acclaimed mechanisms for cellular powering, coherence, homeostasis, electromechanical activities, sensing response are inadequate. That's the reason that I am saying. Now, what we come to the murburn postulates. Murburn postulates fundamentally believe that cells are crowded and stochastic environments, diffusible reactive species naturally arise, and it is unavoidable that you can, stop it somehow, and system level control must be distributed, not localized. Molecules cannot be intelligent per se, but things can come together and they can be intelligent. And very importantly, some point I have missed out in this is cellular medium is not a dilute solution, it's a colloid. Anybody can verify this, but all theoretical treatments in classical textbooks consider dilute solution treatment for biological systems, cytoplasm. So, before I go into murburn concept, I want to just say, I want to pay tribute to Lowell Hager, with whom I worked in late 1990s, from where the journey started. Lowell had found this enzyme, chloroperoxidase, and his idea was the standing principle or mechanism of that heme protein. I went to his lab, availed his fellowship, worked on his NIH project, and opposed his idea. And he was so magnanimous that he accepted it after 6 years. It took him some time, but he accepted it. There you see, my tribute to you. You will live on in my memories forever, Lord. And this is Stanislav Yanev. I was working on drug metabolism. I went back to US. I came to India to do social work. Then I decided that I don't have the ability to get my own following and people will not listen to me. So I wanted to make a big name for myself. I went back to US and I started working on another theme as well. The drug metabolizing liver microsomal cytochrome P450. There, I was in the land of opportunities. Thrice I was fired for holding on to my opinion. That cytochrome P450 is the proteins that metabolize the vast majority of drugs that you eat in the liver. Alright? The liver is reddish brown because of these enzymes. Blood is red because of hemoglobin. Liver is red because of reddish brown because of these enzymes. So these, when I said my project, NIH project, was to minimize ROS production, reactive oxygen species production. But I reported that if I take out reactive oxygen species, the reaction doesn't occur. So what happened? I was fired. They said you cannot continue. So this is the second system. Then I came to respiration, photosynthesis, multiple, then diverse metabolism, vision, mechanophysiology, immunophysiology, evolutionary biology, all that. And this person, Stanislav Yanev, he was also working in drug metabolism. All his work, he had worked through the classical mechanism. But when he saw my mechanism, he said, yes, this makes sense. And he supported me until 2024 when he died. To the two of you, my salute, my thanks. Before I move on, my gratitude and my respect to you. Without that, our society doesn't work. Okay? So, first and foremost, I will talk to you about how I came to this idea of diffusible reactive species can give selective and specific reaction. First is the system that I was working on, chloroperoxidase. That is the fungal protein. Now, what has happened is, usually if you take two molecules, like thiourea and monochlorodiamine, please be with me, don't go too much with the molecular structures, it's not important. What is important for you to understand is, two molecules were given in a reaction. Okay? And when two molecules is given in a reaction, and if I add a chlorinating agent like the toilet bleach, okay, both get converted equally, 1:1 ratio, quickly. Now, but when you give these two to the enzyme reaction mixture, this was consumed first. The sulfur lone pair was consumed first rather than the beta diketone. So, the idea was the enzyme active site somehow selects this molecule over the other, and then when that is not there, this comes. Now what I showed is, you add the bleach in a small amount slowly, you get this converter. That is, sustained release of a radical or a reactive species can achieve selectivity and specificity compared to bulk. It's like I come through the door and if I see Professor Raveendran sitting, I know I like him, I can, but if it is too crowded, my collisions with others will prevent me from meeting him. Otherwise, I'll spend more time with him because I know him more than anybody else. That's the kind of thing. There are even the diffusible reactive species at very small amounts, they have their own philia, they have their own selectivity. That part is what I established. And also here, in the oxygen insertion reaction, you get substrate dependence, whereas the chlorination reaction is practically a zeroth order reaction. So here I said, this is because the radical comes out and reacts. This oxygen insertion at the heat center is through a diffusion diffusion controlled active site reaction. So I could show selectivity and all that changing by pH and chloride ion concentration. So how does this affect marine life and all that? See, chloride ion is high amount in life science, in marine life. So this will impact those ions containing medium more, because this kind of things afford greater amount of reaction impact with the ions. So what is murburn concept? Coming to what is murburn? Now you have a protein. This is a classical mechanism. This is an enzyme. You have an active site. You see the complementarity of the topology. The active site has a very beautiful complementarity with the substrate. So the substrate gets turned over like that. That is, another molecule that looks like this perhaps will not get reacted here, because that's how the active site is. Similarly, electron transfer also, there should be similar topological complementation. Whereas the murburn model says you don't need such things. A reactive species can be generated which can react with a weakly bound non-topological complementary stuff also. So what does Murburn say? Murburn says that mucides are the agents that catalyze Murburn reaction. Extension of classical enzymes, they invoke diffusible reactive species or diffusible reactive oxygen species, or like what Muralidharan sir said, muir burning. It has one electron reactions. Usually one electron reactions are not considered to be very deterministically controllable. So our Murburn burning, like what he said, and then molecule unbound ion radical. Our life is made of molecules, unbound ions, and radicals and radiations. Also, in fact, the components, they interact stochastically or not deterministically. Also, they can give also some reaction or mild unrestricted redox catalysis. This is based on effective charge separation. These are the terms. ECS to give cells that work as simple chemical engines rather than proton pumps, proton motive force, ion channels, specific, all that. These are the key ingredients here. Now, let us go second. This is a diagram made by Danny. He is sitting here. You see, here, inhibitor or substrate, they bind at different or the same place, binding based outcome. So you have highly deterministic, specific redox partner in integral stoichiometry. What is integral stoichiometry? A plus B gives 2C. But here, you see the Murburn reaction. This is gone. Power away. So, you see, in that, the reactive species there is a zone around the enzyme where it can interact with various ions, various molecules, and then that can give altered effects of inhibition, activation, all that. So here you get broad substrate ranges, non-specific redox partnering, and non-integral stoichiometry. Just by one simple idea that non-active site reaction can give this kind of output. Very simple suggestion. So here what happens is, in this theory, you see how the middle one, you see, you don't need complementarity, because the diffusible species in the center, which is shown as stars, they can be produced and they can react with the final target to give the products. So this is what I say is the role of oxygen. Oxygen makes diffusible reactive oxygen species, and that interacts with one electron active species and all that, and there is like a whirlpool from which products come out. So here you have effective charge separation. Oxygen comes in, makes a charge separation, and it can give diffusible reactive species which are not released as such like that. They are released in a sustained release dynamics manner, and then you have these two all together setting up a chemico-electromagnetic matrix which gives the cellular intelligence, and PKs, which is also cellular intelligence, powering coherence, homeostasis, electromagnetic and mechanical activities and sensing. So this is the way simple chemical engine works. So how do you control, like what Sir asked? This is, if you have high amount of radicals, they equalize. If you have small amount of radicals, they will stay. But then there is proton availability, spin locks, all that. These are the ways in which you can control them. So at the outset, before going into any kind of application, you can see from a very conceptual way, oh, it's working. Suddenly I got it. So the first one says different topology molecule. This active site may be like this, but because the diffusible species is involved, different structures molecule can react. Then how different kinds of molecules and with different moieties, you can get different kinds of reaction. By the same enzyme you can get. So earlier this was not explicable. And then the kinetics that you observe, that is, you have Against maintained kinetics, you have Km is equal to Kd plus K2 by K1. If you are an ardent student, you will know. That is, Km is a reaction constant, but Kd is a thermodynamic binding constant. Then how is it that sometimes Km becomes much orders lower? That's because Km is reflected by the diffusible radical species. So it can explain how you get various types of kinetics. This is very important. It has got medical applications. That is, in certain reactions, you might not get in certain dose administration 10 milligram drug. You might not get an effect, but 1 milligram you can. Why? Because at 10 milligram you have larger amount of radicals. Whereas at 1 milligram you will have very less. When that very less amount present, it is stable. Too much radicals, they all fight with each other, react with each other. They will destroy each other. So radicals, when present in a small amount, it serves. Just like in society, just like in chemical reaction. Alright? So, lower amount, say, I am not advocating homeopathy here, but then some drugs can be more effective at a dilute concentration. Then variable stoichiometry, there is a lot of different fights. We get 2:1 ratio. Somebody says I get 1.5:3 ratio. All that, 1.5:3.2. Non-integral ratio. All that can be explained by this new idea. Most importantly, how cellular electricity, transmembrane potential. That is, the classical theory said that 3 sodium pumped out. So positive ions were pumped out. Rather, this theory says oxygen comes in, takes electrons, becoming negative inside. So phospholipids contain this negative charge inside. So inside will be negative. You don't need to pump anything out to make the electrical activity. Alright? So here is the way in which the pumping is replaced. NADH gives reactive oxygen species inside, and then that makes ATP. Alright? So here you have in the proteins, colloids, they can bind to ions and co-solvation. We will show that potassium is more soluble in the cellular medium because of carboxylates, proteins, phosphates. Thermodynamically, potassium is greater stable and the presence of diffusible reactive species inside the reducing environment thermodynamically favors potassium. It's not a pumping. People did not understand the thermodynamic foundation. That is why. So that's why the bigger ion of potassium is inside. So this theory predicts that DRS levels should correlate with function, lipid phases should modulate redox functioning, buffering, and non-integral stoichiometry should be usually noted, which is the case actually. So we don't need pumps, and this says accepts colloidal nature, and it also, these are radical reactions which can be quicker. Genetic control mediated or binding based effects are relatively slower. You cannot explain the kinetics. So this is beautiful in that way. So one example I will come to here is respiration. The most fundamental process of all living beings, all living cells, particularly aerobic cells. Aerobic is the ones that use oxygen. The classical theory, what Sir was talking about, is this. That is, there is the membrane, mitochondrial membrane. There are these large proteins, complex 1, complex 3, complex 4, that is complex 5. Now, they are supposed to be serving as part of electron transport chain, and there are diffusible species like ubiquinone or coenzyme Q or cytochrome C here, okay? And that is supposed to relay electrons between these fixed stops. It's like a manufacturing line. One electron goes, two electrons go from here like that. So that, and while that is happening, protons are being pumped. So protons can't be pumped because protons are too small to be held like that. There is no real gradient observed in physiology, and these are the reactions, and these are the kinds of ratios. So this is the structure of mitochondria. It's something like a hydroelectric power plant, where you imagine there's a water at a higher level, it comes in, it sets the turbine going, and then you have somehow that is pumped from that. So protons are like water here, they are pumped continuously, and ATP is made. This is the classical theory. Alright? So what is the murburn theory? Murburn says oxygen comes in, all those electron transport chain molecules, proteins, they make these species radicals. Radicals attack phosphate and ADP. ADP is bound at those proteins that are on the membrane, and so the probability of the radical meeting the ADP and phosphate is high. If the protons are low, the radicals won't become water. They can react with ADP and phosphate. That is a beautiful reason why bacteria are non-micron. Because only then protons will not be available. Otherwise, what happens is the radical will quickly take proton and become water. If proton is not available, it takes millisecond times to come in. Before that, it can react and make ADP. So these reactions are simple bimolecular reactions. Simple bimolecular. It doesn't require something 4 or 5 protein, each protein being made of dozens of genes to come pump or control across multiple phases. These are simple bimolecular reactions. Okay? This is the murburn one. So this is the overall layout. That is, complexes 1 to 4 use different molecules. Now, sir, ask why are they present? Now, complex 1 can use NADH, succinate dehydrogenase can use succinate, complex 3 can use so many things in the vicinity, and complex 4 can use peroxide. It depletes the peroxide to completely to water. So all of them generate DRS, and they make ADP and they react with ADP and phosphate to make water. And then the ATP formed is taken out by porters and water formed goes out to porins. And when this is happening, protons from outside come in. So these are all directional. There is no cyclicity in this. Everything is directional. Also, in the earlier one, you need to arrange the complex 1, 2, 3, 4 in a particular way, all that in particular ratios, which are defeated by the super complex structures that are available now. So here, you don't need any such arrangement. This new theory explains the multiple modality that you see mitochondria in. And most importantly, we showed in lab, you take ADP and phosphate, add superoxide, you get ATP. And this reaction is inhibited by cyanide, experimentally. Very simple reaction we showed. And not only we, Kathleen Naylor had shown that in a mitochondrial suspension, if you take xanthine oxidase and xanthine, you get increased ATP. Xanthine and xanthine oxidase give superoxide. It's known. So also, there were some other lady scientists who had shown like Mildred Pond. In 1950, she had shown multiple O atoms incorporated into ATP. That can only occur through radical reaction. So what are the evidence we have? More NADH and more oxygen increases diffusible reactive oxygen species. Increase in ROS. This is Nicholas data from AG Cell. He is the guy who has written bioenergetic textbooks. You see, when ROS is present, only when ROS is present, you see ATP formed. Only when ROS is formed, you get that electric transmembrane potential going to 200 millivolts. So, it is the ROS that is doing. ROS is not a byproduct. They always thought ROS is a byproduct. ROS is not a byproduct. ROS is a fundamental agent. So, ATP production is not seen without ROS. And complex 1 to 4 generate, all these complex 1 to 4, they generate reactive oxygen species. And all these pendulous regions are ADP binding sites. There is no role in the classical field, at least for complex 5 there is, but in the other ones there is no role, definite role. Okay? So I will come to two specific proteins. Some of you who are biologists can see this, please bear with me. Here is some chemistry, don't get lost here. Here, this is complex 1, perhaps the most interesting protein in the electron transport chain. This is the prokaryote, this is the eukaryote in our system, this is in the bacterial system. They are the same structure. They have multiple iron sulfur centers and flaming at the top. And this part is supposed to be the proton pumping, and this part is supposed to be the electron transport chain part. So they are disconnected, physically disconnected. And not only that, the electron transport is supposed to occur in microsecond time scales, whereas proton pumping is supposed to occur in millisecond time scales. There is no spatial, there is no temporal connection. And also, the murburn theory beautifully explains why NADH can give that it gives only one proton but gives two electrons. So, we want a proton deficient system wherein the radicals can be established. All these are deliberate ways. Why should you have all these answers? Why should you have all these redox centers here? If the classical electron transport chain was true, you should have redox centers in the membrane. So when the electron goes through here, protons could get pumped out. Most importantly, this bioenergetics, the classical reactions, if you see, it's endergonic. See murburn reactions. They are beautifully exergonic. They have high drives. If you see all medical biochemistry or standard biochemistry textbooks, the delta G values will be very small. These are very exergonic reactions. On all these prokaryotic to eukaryotic, we showed, Abhinav showed that they have high conservation of ADP binding sites. So these are the actual fundamental ADP binding and ROS reacting agents that we make. So this is beautifully shown, and then complex 3. In the classical theory, there is no role for this subtension. We showed ADP binding sites here, and all these Peter Mitchell said OQ cycle, Q cycle. All these genes and answers, they are all solvent accessible. If there is any wiring, you would not imagine that. There is no way this can conduct any specific wiring. This is random, evolutionarily optimized way of using reactive oxygen species and coenzyme Q, that is ubiquinone here. This is in murburn system. This is a radical scavenger in the membrane. It doesn't need to take from one place, from complex 1 to complex 3 like that. This is immobile practically. It's too big. And so what it does is, there are different kinds of lengths of these things. They are packed in the membrane. And why? So the larger one is the more predominant one. If you had this serving as a transfer agent, you would imagine the smaller one to be preponderant. But no, otherwise. So this is the new theory. You can see, like earlier, all the murburn reactions are exergonic. Thermodynamically facile, kinetically facile, agrees with the structures all, and agrees with the inherent nature of mitochondria. This is a work that Danny and I, we had predicted in 2018 in biomedical reviews that thermogenin, the protein that is supposed to generate heat in the body, in mitochondria, we studied the protein, whatever structure was found. We said it will have high lysine and arginine in the interfacial area that will enable the reactive oxygen negatively charged react. If you have lysine and arginine, they are positively charged. So they can move things better. They can facilitate proton dynamics better. So if you have proton dynamics facilitated or movement of anionic species, they are also called anionic transport agents. Now, if they can move it, then heat will be high. So we predicted that the structure will be like that. True to our prediction in 2024, it was revealed that it did have. So we sent our paper, letter to the editor to Science. They published it online. They can offer rebuttal. After 2 months, they took it out without even informing us. The editors didn't offer, or the authors didn't offer any rebuttal. That is a Nobel Prize winning people's group. So that's why we should have some mechanisms to advocate our own side. So the classical theory required fatty acids to say how the classical theory said this proton gradient is dissipated using fatty acid. There were multiple theories. We don't need all such theories. We can explain heat formation in body beautifully based on radical reactions. Their kinetics are fast. So when rocks react among themselves, they make water and produce heat, and that leads to thermogenesis. Beautiful structure and function, beautifully explained. So now this is the macroscopic view. I still have about 10-15 minutes. We finished in 1 hour. So the idea is here that here it's like a grill. You know, some heat source happens and some material is placed and some reaction. That gives a very simple chemical process where you can draw analogy. That is steam, like you have a combustion reaction, carbon dioxide, or all that you can find analogy. Unlike in the classical theory where you had redox process disconnected from the phosphorylation process, this is a directly coupled bimolecular reaction that's overall highly facile. So it explains transmembrane potential, thermogenesis, Turgor movement of water explains homeostasis. So this is a fortuitous assembly, independent and parallel functions, agrees with the architecture. Overall, this is very simple. So whole perception changes. Fatty acid giving rise to toxic signaling and all that in heat and all that here. From here, the whole chain of understanding changes. So this is a holistic idea. This is something that I was talking to our sir, Vasudevan sir, only 2 months back we named this. That is, Krebs cycle is another reaction that is happening in the cell. After glycolysis, acetylcholine coenzyme A, it is making the NADH. Now here, I analyzed the thermodynamics of this reaction, whole cycle. I found it to be highly endergonic. It is 10 to the power, the K equilibrium constant comes to be 10 to the power minus 127. 10 to the power minus 127. No way Krebs cycle reactions can occur if the murburn reactions don't occur. So the carbon dioxide releasing function can occur only with oxygen. That is beautifully explained by the murburn view here. Here, it requires NADH to be made. Krebs cycle cannot work without this happening side by side. So rather than a parallel reaction system, this is like a cauldron where both occur simultaneously. So the thermodynamics, kinetics, feasibility, everything in mitochondria, this is conclusive argument. I gave it to ChatGPT, I gave it to Cloud. They finally said, this is too much for anybody to escape. So this only 2 months we have not yet published it. So this meeting has several unpublished release of ideas. So that's what a seminar is supposed to do. Meeting is supposed to do. So this is something somebody should have come online. He is not online. Paul Gagnew's idea. I went to Bulgaria this March. I was invited there to present. And this is, he was studying the dimensions of mitochondria. And somehow, here it is mitochondria thickness, mitochondria length. He presented one data and he said, there is an area, I don't find mitochondria. Very simple. Here, pi r squared is the volume of mitochondria. If your thickness high and length also high, the volume is high. When the volume is high, protons are more. If protons are more, it cannot function. Very simple. Very, very simple. So he bought it. But he didn't come today online. He said, then red blood cell, another application. I told you how can red blood cell sustain. Red blood cell has hemoglobin, perhaps most abundant globular protein. It's a tetramer. It has 2 alpha and 2 beta monomers. The classical theory has nothing to do with beta monomer. It is just like alpha. But you see, 2 beta monomers, they have got, this is the way oxygen goes in, and the other side has a small hole. In the tetrameric tense form, Abhinav showed that it binds to ADP, and when iron 3 releasing superoxide or iron 2 releasing oxygen is the same. Both can be possible. So when it releases superoxide, it can make ATP. This alone explains how you can get 3.5 millimolar of ATP in red blood cell. So you can get how maintaining high amount of phosphate always inside gives more potassium inside, homeostasis and all that, energetics. So overview, we are coming towards the end of my talk now. I will finish it in 1 hour as expected. Overview. So now we understand why the rod cell has high packing of lipids. Mitochondria is like this. Same thylakoids, cyanobacteria, liver microsomal, all these places where reactive oxygen species are high. Why do we have high lipid packing? Why? Because water should be minimal. If you have water, you will have protons. So that is why. Then we showed that in all places murburn reactions occur. Then we showed that post-translation modifications are not just post-translation, not just at genetic level, at a metabolic level also you have multiple kinds of impact by murburn chemistry, radical chemistry. That's what is shown here. Post-translation. So you come to an overall macroscopic and molecular and conceptual understanding where molecular level, diffusive reactive species interact with iron and all that, and they can make chemical processes. At a molecular level, these chemical processes can give charge, heat, pressure, because water is formed, gives pressure. So all these things like a simple combustion engine, you don't need pumping, you don't need molecular level intelligence. So it works at a systemic level like that. And so at a conceptual level, short-term idea is murburn concept, stochastic, and then deterministic information long-term you give set. So here, diffusive reactive species, effective charge separation is the fundamental foundation. So now, our understanding of overall conduction of, earlier you see, you would imagine it's like a classical orchestra where everyone is given a piece to be played in a given time. But then a jazz band works in a different way. People have an idea and they just play along based upon each other. This is the new idea that you get. It's not that deterministic. So now here you understand why oxygen became so important. Oxygen can directly enter into equilibrium compared to those sulfate, nitrate reducing bacteria. Here oxygen, you can exploit the full minus 400 to plus 800 millivolt full range and you can get that. The ions are electrolytes and lipids are proton limiting environments. Proteins they have active site specificity. Okay. Carbohydrates, why carbohydrates form the basis of nutrition? Because OH groups, water, hydroxyl, that is the way it has to go. Nucleic acids have phosphate and OH groups there. Why? To prevent the loss, to attack the bases. So oxygen needed for generating ROS. So from this, you can have stochastic, spontaneous interactions. You can have emergence of biological intelligence through weakness. So here's a new concept where spatial probability. Murzymes, these were the primary enzymes, and classical enzymes are when the transition state and spatial reaction sites are minimized, you have the classical enzyme. The Murzymes are the largest set of enzymes. Murburn does not negate classical mechanism. It complements and extends them. This is a misunderstanding that people have. Alright? So, here is a basic idea that people can very easily relate to. That is, if you find a knife in kitchen, it is also associated with cutting board and knife racks. When you find a knife in kitchen, you don't say, knife is not to be used. Similarly, if you have antioxidant enzymes like catalase, peroxidase, superoxidase, speedase, it doesn't mean that the antioxidant species are bad. It means that we need it, but it's like knife, it has multiple problems. So, it can give order, it can give chaos, it can give both. So, these are the elixirs of life. They explain, science is about, physics is about energetics, kinetics, mechanism, structure, distribution, evolution, probability, organization. All this Murburn explains better. Only thing it doesn't explain is the stigma or aesthetics. What if, Sir said, how do you get the control? There are control mechanisms, but then it is in a sustained way. We have to understand how all this happened. So, it is, we studied in school that the difference is like this between combustion and respiration. I say respiration is also very much like this, and in the Murburn school of thought, ATP is important, but it is not the complete. So, very important, why do you die? Because fundamentally, you are like a cloth that is dipped in oil and set fire to it. Now, the oil burns, the cloth will also burn. But the oil burns first. So you are born, you will use oxygen like this, you will die. It is destined that you will die. It is not a toxic side products. Murburn dictates that you die because these are the imperative agents in life process. So, Austin has this Dr. Jekyll and Mr. Hyde or the Batman comics two-faced, you know. It is not the classical theory says oxygen goes and binds to cytochrome oxidase to receive 4 electrons and 4 protons to make water. It's not the way. The workers of life doing chemical, thermal, electrical, mechanical, all these works are the diffusive reactors. They connect things, they react, they destructure water matrices. We are composed of 70%, 80% of water. All this structure, restructuring, when we move this, all this is happening because of water activity and not just protein. We have to see beyond all that. So that's the role of oxygen. That's what we have to start seeing now. Alright? So, impact. Impact, you see, this video has to be played. Can somebody play this video? First, it's getting recognition. Is there audio? Okay. That's George Charlacov. I was invited there. Thank you. That's George Charlacov. I published first this Murburn model of biogenetics in his paper, 2017. Next year, Scopus Index 1, PubMed 1. Next year, the very next year. But every year after that, he has put Murburn on the cover page. He was supposed to come online, he is not able to. He gave the message, perhaps we'll play. So, what's in it for clinicians? I say that Murburn is a, perhaps in due time, it will save more lives than any other quantum of information. It will prolong life. Now we are 60 to 70, we will live to 100 in 20, 30 years, and it will enrich our life more. So, I don't want to go on now because we have to have discussions also, it's already 1 hour. So, here you see what is the impact. After the publication, proton motive force and chemiosmosis is going down, mitochondrial work is going up. Recently, just this month, we saw pharmacology, it was 20 impact factor. Chinese people, citing 5 papers of murburn in mitochondrial metabolism. So this paper, Chinese group, again 18 papers on murburn. Alright, so another one on hepatocellular carcinoma, 5 papers on murburn. Alright, so this person has given a message, Marko Vitas, we'll play it later. So I would end by saying, we in our culture, we called digestion assimilation process Dahanam, Dehagni, Kayagni, we call it. Why? We have our body filled with. I think it is not really great to dismiss all ancient ideas and terms just like that. Shante Abnam Riyate. There is a fire burning inside. They must have had some intuitive idea. It's like burning. That's why they used it. I also say that. But I got to know about it only now. So this is my favorite comic by Bill Watterson. It's a magical old world buddy. This is a 6-year-old boy talking to his stuffed tiger. He says, let's go exploring. Why cameras? Always, if you are young budding scientists out there, don't take anything anybody tells you unless you try to understand. Alright? When you try to understand, when you ask questions, then is where you start becoming scientists. These are questions. These are all questions I have prepared. Okay. Thank you so much. I thank before I I think Professor Babu, Professor Vitas, Rajendran next session. He can call him here also anytime. Now, Professor Babu, he has been with me for a long time. Professor Babu, can you please say, Professor Vitas?
Yeah.
Professor Babu, please go ahead.
Yeah. Can you hear me?
Yeah, we can hear you.
Okay. All right. Thank you for inviting me for this special presentation. It's kind of late here, because Earth is a round object. Time here is 2:50 a.m. But I kept awake so that I could listen to Murli. I have known Professor Murli since the early 2000 when we were working as postdoctoral researchers at the University of Illinois. And I have seen firsthand the inertia shown by some scientists to have an open mind to ask logical questions about these classical models and how they compare with the murburn model. However, the past couple of decades have witnessed a steady progress in the research and the reach of this model, as Professor Murli has correctly described. It's not just a simple model or a new theory that explains some few processes like cellular respiration. It indeed is a paradigm shift with far-reaching implications for our understanding of life and its evolution. So I would like to make 2 important points here. One, the scope of murburn model is very broad. Professor Murli has already shown that it can be applied to a number of phenomena. He has explained the mechanism of vision, how very little amount of cyanide can kill a person, and so many other examples. So a lot more research can be done in this field, and many physiological processes can be looked at from the perspective of murburn model, and this will certainly lead to new advances in both basic science as well as applications in the field of medicine, etc. Now, I'm a physicist, so I would like to make one important point. We all know that living organisms, starting from, say, the single cell forms of life to the apex predators, they represent thermodynamic systems. Thermodynamics is one of the oldest and most fundamental branches of physics. The laws of thermodynamics are fundamental laws of nature. Living organisms represent complex thermodynamic systems. The degree of complexity increases as we go from, say, single cell organisms to more evolved animals. Now, as far as thermodynamics is concerned, no matter how complex a thermodynamic system be, it must still obey the fundamental laws of thermodynamics. The murburn model is a theoretical framework built in accordance with the basic laws of thermodynamics. So that is one of the most important reasons why this model is going to prevail and emerge as a basic principle of life and its evolution. Thank you.
Yes, thank you so much, Babu. And you will witness also to the fact that Lowell said that these people would not accept it. You remember that we were in Harvard?
I remember that very well.
Yes, thank you so much, Babu. It's late. I know, please. Thank you so much.
Yeah.
Thank you. Yes. Professor Vitas, please. Dr. Vitas. Marko Vitas, can you?
Yes, I can hear you.
Yeah.
Good morning.
Yes, good morning. Vitas and I, we got to know each other after my research, and we interacted over. He's an evolution specialist. So we know each other only through work. We are not collaborated. He's an independent researcher. Please, sir. Please.
Thank you very much for this very interesting lecture, beautiful lecture, where the whole complexity of murburn concept was detailed, and I would like to point out that iron sulfur are very ancient enzymes, and this murburn concept will contribute to understanding biocatalysis and evolution of biocatalyzers, and also consequently would impact our understanding of origins of life. So may I ask you a question?
Please do, Professor Vitas. Dr. Vitas.
I would like to ask you, what is the most provocative part of your concept in your modern concept? What is the most provocative which you found difficult for a broader scientific community to accept it?
The most provocative is the one that Professor Vasudevan said. The third part, actually the second and third part are part of the same question he asked. He said, how do you achieve control? Because you have a radical, oxygen radical. Currently it is seen as something that creates chaos, creates diseases, is resulting in aging, all that. I don't say that it doesn't, but then it has a good side. So to get people to go beyond the aesthetics, beyond the stigmatic perception that prevails, that these reactive species are bad. I don't say they are not. I don't say they are not. I mean, I'm not getting the logic. I say they could be bad, but the case is that everything in life has got a context. It has to be in the right amount, at the right place, at the right time. Three things have to come together. So, what I say is reactive species at the right place, at the right time, in the right amount is good. It can give practically zero thought reaction. So something changes, this will keep it stable. You want something to change with change, but you want something to be stable of ideas. They will naturally see that this is more conducive to evolution, more conducive to probability, more amenable to structure, function, distribution, thermodynamics, kinetics. And that's what we try to understand life. Thank you. Anything else, sir?
Well, thank you very much.
Thank you so much for staying online for so long. Sorry that we got a little delay. Sorry.
Okay.
Thank you. Yeah. Professor Babu was from Western Illinois University. He's a professor of physics. He's a university. I think those people who are online, please excuse because it's already become late. Our sir, please.
Mohan and Rajendran will speak in this.
Next, yes.
Others, if somebody want to ask,
Who else is online? Mahendra Kavliya can use another session. Dr. Hussain is my collaborator. So those two were guests, they have not collaborated with me. So who else? I think Professor Sumar also can. Mahendra, hi. We can talk sometime later because I think it's already 1:30 and people will have to go for lunch. We will hear all. Morning, sir. Please bear with us. So, if anybody wants to say something, they can say, but otherwise, it's okay, I guess.
Sir,
Yes,
yes,
please go ahead.
Hi. So, sorry, I know that it's late, so yes, very nice. The second time I'm listening and the lecture last time was in CPCRI. I have one quick question.
Please.
Did you do any, first, initially I didn't listen, so that's, did you do any experiment using isolated mitochondria? And the one you might have seen, there is a classical experiment where isolated mitochondria just they put it in two different water systems by changing the pH and where vanillamides they put and they simulated an experiment and they put thereby ATP without substrate actually. So did not do any similar kind of experiment so that you will add more support. And as a being a teacher who teaches this particular topic for my biochemistry student, being a biochemist, so which direction? Of course, it's a very novel thought, no doubt in that. As Vasudevan sir mentioned, I think I didn't hear it yet, and the control of this ATP synthesis is becoming a big question there because mitochondria is very sensitive for the free radical. So cytokines will be going out and cell will die if there is a imbalance in the free radical content of the mitochondria. So that's why I have this particular question. Of course, we have a lot of questions we will discuss another time. I know that it's very
No, sure. This is a very important question because Jagendorf's experiment with chloroplast, that is what triggered all this. When Peter Mitchell proposed that proton difference gradient is what gives rise to ATP, they did not really understand the thermodynamics behind it. And they did an experiment and it was false interpretation. I'll tell you what. Now, H plus ADP plus PI plus H plus gives ATP plus H2. That is the actual equation. So in that experiment, what they did is that they gave surplus H plus in this surrounding. So that is thermodynamic because H plus contributes in this equation 452 kilojoules per mole on the left side. When it gets on the left side, you have delta G formation calculated by the right side minus left side. So 452 joules on the left side you get highly facile ATP synthesis. But that has no relevance whatsoever to physiology. Because in physiological system, there is no pH gradient. Inside is the same as outside. So now if I take a lion, put it in a circus cage and make it jump through some fire hoops, and I say, in reality, it is in the jungle also it feels like that it has no relevance. So, Britain Champs very clearly showed in two Nature papers when Peter Mitchell proposed that this proton gradient is what is given. He showed very clearly there is no pH gradient. I don't have the expertise or the experimental setup to do, but I can easily rationalize it. My cover page credited paper in Progress in Biophysics and Molecular Biology clearly explains these experimental results. Why the bioenergetics community went on the wrong side? Because H plus contribution was not really taken. That's the reason. So that doesn't have any physiological, and they forgot one thing. They did an experiment with rhodopsin and complex 5. And after that, first just with mere mitochondria they did, and then they showed incorporating rhodopsin like induced proton pump. But they did not realize when they lacked complex 5, Rhodopsin alone gave ATP, 20%. That data they completely refused. So murburn is a more complete explanation which explains what complex 5 does. Complex 5 uses ATP and opens up the internal. See, when you have a stove, chula, when it gets stuck after a particular time, you need more air to come in. That's what it does. It opens the, uses ATP, opens it, lets outer protons come in. When protons come in through solvent, then they bound ATP on that, use phosphate, and can also make ATP with that. So that explains how ATP is made in the ATP.
I think you have a lot of reception of this anyway.
Yes.
Okay, thank you.
Wonderful question.
We have one last question from you.
Anyway, questions have prizes at the end of the day. Krishna Master.
No, but there are no questions every lens.
Please ask questions. How many of you have studied this? Respiration? Yes. I taught you guys. So the boy raised a hand and the girl asked him the questions. What is the purpose of ETC? I showed you, perhaps that would have answered. The aesthetics control, I posted a slide, it had some concept.
And one thing is really complex and the other thing is very straightforward. Then usually we take this straightforward theory. The moment that he does a few years back we met and we explained that the simplicity of the theory is what attracted me first place. That it is so natural, a natural concept is there. And that is what attracted me, and that is why I took it to my textbook also as an alternative theory. Now, secondly, the photosynthesis, see, the main point I want to put is that the RBC, even from my undergraduate days onwards, I was asking myself and all my teachers and colleagues, etc., how the RBCs are there for the 120 days lifespan, whereas there is no enzymes are there, I mean, not enzymes are there, but there are no DNA, there is no protein synthesis, no ATP synthesis, the whole the ATP synthesis machinery is not there as per the current theory. The model is working like that, and it's not really an answer for that. But this is an answer, that the direct answer that you can give. You have to experimentally prove that is another matter. But at least a theoretical concept is coming here. That is a great point that I take it as a point that is not for this particular concept. And lastly, I want to prepare that you want to take one concept to Mullany's concept. Because otherwise, Mullany's concept. Because otherwise, after 10 years, some Westerners will think that it is a public system concept.
Sir, yes, sir. Sir, I wanted to live beyond my name. So I have worked my whole life, 27 years now. He said, no, only one. So, but then I have commented, sir, when Peter Mitchell can say chemiosmotic, which doesn't have chemistry or osmosis inside, and he can all say that Mitchell. I have 5 reasons to give murburn. Molecule, unbound ion, radical radiation. My unrestricted, I taught a lot. My name, today I am here, tomorrow not here. When I was in 12th class, sir, I wanted that I would read all these name reactions, Kasaragode reaction, MCDF reaction. I wanted to have something new. We have kids, we have work, then we have ideas.
I just want to have something. I must admit that I haven't understood much of this. But one thing I just wanted to comment on is some of these things which you say at the molecular level. I think we still have to go down to the molecular level. We are still talking about the microscopic words instead of molecular levels. For example, I still remember one paper by Niels Weber, the full German spectroscopy paper. we have actually discussed about there is a lot of coordination chemistry and so many things which are important. But he was actually talking about the movement of this, the ion oxide system. And he was talking about the thing going from square planar to pyramidal symmetry.
Yes.
You can actually see that molecular model. There is actually a lot of coordination chemistry that is involved in the oxidation, deoxygenation, and all these things that we learn about, which I do not see in this kind of a microscopic perception. Probably that is what will help to get added up in the coming years.
Absolutely.
I follow him that I could not follow any of the things. Naturally it is complex, but there are certain main questions that to ask you. You were talking about the size of a proton. Okay? The size of a proton for negating something. That is positive side or negative side. For example, size of a mitochondria. Okay, up to that level it is okay. But if you try to explain on the basis of the size of the proton and arriving at a number 6 there, I don't think scientifically, for example, the size is considered for a particle. In a proton level, you say that, okay, this is a particle and it has a size. It is difficult for me to immediately think that proton as a particle. Okay? So, relating to the level of a proton, electron, their size, to explain and observe the phenomena that is getting too congested, how it is possible, etc. What is the rationale of this type of a correlation? This is one question from my side.
Sir, that size
is a question.
Wonderful. So the size is not the one used for calculation. The calculation is based on thermodynamics. The dissociation
number, etc. You did a multiplication.
Yes, sir. That water dissociation equation, the pH measurement very clearly says H plus concentration is 10 to the power minus 7 to 10 to the power minus 8. So the molecule, the concentration is that much. So you multiply it by the volume, you get the number of actual protons that can be theoretically present in that volume. So that's what is the number. So the theoretical calculation is absolutely right. In the sense, the dimension is used for something else. The dimension of a proton is known. It is approximately 1 femtometer or something like that level. Now, but then protons don't exist like that in reality. Protons exist as hydronium ions, Zundel, Eigen, different levels of hydration. Now, but when biochemists or biologists use protons, they speak like protons stand there H plus, the glutamate takes H plus like that, they show it like that. But if they exist as hydronium or their hydration is important, they should also try to talk about the hydration energy, the size of the hydronium ion or Zundel ion or Eigen ion, all that, which they don't do. They conveniently pass it on. My point is, the size of proton is so small, individual, it is impossible for ions, larger atoms to selectively bind it. They say selective binding and pumping. That's what I'm questioning. Because
that's kind of different description altogether. I don't think there is any proton living as such. So when there is nothing of that sort, there is no scope for such discussions. But the proton transfer in water particularly, there are beautiful ways that people are shown. You will give a proton here and take the proton there. Almost like a Havala then.
yeah, Broca's mechanism. It is called Broca's mechanism.
Yes, that's right. In biology, there are a lot of beautiful things like with respect to proton or electron transfer and how the chains, these proton chains actually assist in that. Anyway, I think it was a wonderful discussion and we will end up now for the time being. I think the lunch is waiting for all of us. And thank you, Dr. Murthy, for that.
Thank you.
Beautiful presentation. I hope you have a beautiful concept that you have put forward and you will have the questions that comes out of that and answers that are coming out your side or somebody side in the coming years. Eventually, science things get ironed out over a time, period of time. So thank you very much for our question.
Thank you, sir. Thank you. Thank you. Thank you, sir. Thank you, Mr. Sir. Thank you.
Yeah, just a second, remark I would like to make. Kids are playing the building blocks. So if the building block is having a 10-pair length block on, so there are different mechanisms. So in nature also, like we have seen only the 10-pair blocks, there are much more ways to resolve the things what is happening. But when we study science, we generally try to look into the significant correlation with the cause and effect factors. So whatever 90% goes, we think that it's well and good, so we establish such theories. But in all the cases when this 90% factors holds good, we can standardize those things and look for the 10% thing. So same way we have to still work on refining the idea.
Definitely.
Standardize those things, bring this 10% and then look into more possibilities doing it. So all the building blocks can be properly inserted. I also admit along with my coaches, I also partially understood what you are saying. There are different mechanisms of resolving some issues. My biochemistry starts and ends with the nature. So, not near that. So, thanks for this wonderful lecture. I thank the panelists joining with us, Professor Raveendran, Professor Vasudevan, Professor Venugopalan for chairing this session, and Manoj for opening up the ideas on murburn.
Thank you so much.